This video will talk about introduction of multivariate molecular profiles to the CIViC data and curation model. Combinations of mutations in cancer are of increased interest as our understanding of the clinical relevance of certain combinations of mutations increases. We now recognize an aggressive subtype of diffuse large B-cell lymphoma called double-hit lymphoma, which makes up about 5% of cases, and is characterized by rearrangements of MYC together with either a BCL2 rearrangement or a BCL6 rearrangement. Also, there's interest to find patient populations that may be more responsive to immunotherapy. So, for instance, one combination of variants with good potential for immunotherapy treatment are non-small cell lung cancer patients with PD-L1 expression, but not the EGFR mutation or ALK fusion. So here you're defining a select population of PD-L1 expressing patients without these well-characterized and very strong lung cancer driver mutations. So let's take a brief review of CIViC and then see what's changed in order for us to be able to start to incorporate these complex combinations of variants into our data model and workflow. The old CIViC data model is based around individual genes, variants, to which we associate evidence and assertions. So you could go to your favorite gene, see a list of variants associated to the gene, search for your favorite variant, select it, to come to a page full of curated information about your variant and the collection of evidence items which are associated with the variant. These evidence items are the hand-curated fundamental units of CIViC. In this case, here's an evidence item describing some preclinical work in lung cancer cell lines containing the EGFR L858R variant treated with afatinib, and the investigators find that these cells are sensitive to afatinib treatment. Now let's take a quick schematic look at the data models. So in our old CIViC workflow model, we were going from gene to the variant, then selecting from the evidence items associated to that variant. In our new CIViC workflow model, we introduce molecular profiles. In the workflow under this model, the user can come to CIViC, check out their favorite gene, and then they'll see a set of molecular profiles associated to that gene, and now evidence items are associated to the molecular profile. Note now what's different in this picture. See how the first molecular profile is associated to two different genes in this picture, whereas in the earlier data model, a variant was only associated with one gene. Now, with the addition of molecular profiles, CIViC has not done away with variants. Instead, molecular profiles are comprised of variants. We break molecular profiles into two different types. First off, simple molecular profiles are comprised of one variant. This then takes the place of the old data model, where a single gene variant can be associated with multiple evidence items. Now simply, the evidence items are associated with the molecular profile, which in the simple molecular profile case is comprised of a single variant. The more interesting and novel addition to CIViC is the complex molecular profile. The complex molecular profile can be comprised of any number of variants. Importantly, these variants can be associated with different genes. Another thing to note is the way that the variants within the complex molecular profile are tied together via ands or ors. And now it's the whole complex molecular profile that gets associated with evidence items. These evidence items are curated out of the literature and give clinically relevant information, which is associated not with individual variants in this complex molecular profile, but instead they give clinical evidence for this whole complex of variants. Let's now take a quick look at the new interface in CIViC, which contains the molecular profiles. We see now that the interface contains entities such as variants, genes, evidence assertions, and we've added molecular profiles. To further take a look at how this plays out in the new interface, we'll go ahead and check out a gene, say BRAF. And right off the bat, we see that the gene interface is very similar to what we're familiar with one new difference, which is that by default, we're being shown molecular profiles that the BRAF gene is involved in. We can open this up, load up the molecular profiles, and we see now the list of molecular profiles in which BRAF is involved. The majority of the list here are simple molecular profiles, which are constituted from one variant, such as BRAF V600E, BRAF mutation, BRAF V600K. But at the very bottom, we see some interesting new things, such as BRAF V600E and EGFR L858R and EGFR T790M. Before taking a closer look at that, let's click over to the variants view and open it up. This is the old presentation of the way that variants were presented in CIViC, including V600K, V600G, and the other familiar BRAF variants. So if we click back over to molecular profiles and choose BRAF V600E, when we open it up, we get to the BRAF V600E molecular profile page. And remember, this is a simple molecular profile, which is constituted from one variant. We see the familiar curator-generated description for the role of V600E in cancer. We see the variant coordinates, which are hand-curated, and information pulled in from other databases such as Allele Registry and ClinVar. And recall that evidence items are now attached to the molecular profile. Indeed, we see a lot of evidence items associated with V600E. If we click on one briefly, we see a predictive evidence item supporting sensitivity response to vemurafenib from a clinical trial. If we click back then and scroll back down, we also see the assertions that BRAF V600E is a part of. Now let's contrast this BRAF V600E molecular profile to the V600E variant. There are multiple ways to do this. We could click the back button on the browser, or we can just click right here to get back to the BRAF page. And we see BRAF again and are shown the molecular profiles by default. Let's go to the variants and find V600E. Let's look at the V600E variant page. We're still given the V600E variant description in this view as well, along with coordinate information and information pulled in from other sources. And crucially, at the bottom of this variant page, we see all of the different molecular profiles that contain this variant. At this point, we've put in 4, the simple molecular profile BRAF V600E, and then some complex molecular profiles below it. Scrolling over enables us to see them. Here, for instance, is a molecular profile containing BRAF V600E and EGFR L858R and EGFR T790M. Let's now click on this complex molecular profile. On this complex molecular profile page, we see the makeup of the complex molecular profile. In this case, BRAF V600E and EGFR L858R and EGFR T790M. We're also given information on each of the variants that constitute the complex molecular profile: BRAF V600E, EGFR L858R, and EGFR T790M. And recall that evidence items are attached to molecular profiles. And as we scroll down, we find an evidence item attached to this molecular profile. Let's take a quick and more detailed look at this evidence item. We see that this evidence is still in the submitted state, since it's not yet been reviewed by an editor. It looks like this is a predictive evidence item type supporting resistance to the drug osimertinib in lung adenocarcinoma. This is a case study from this source publication. And again, the molecular profile is BRAF V600E and EGFR L858R and EGFR T790M. Let's take a quick look at what was curated out of this paper. We see this is a case study involving a 42-year-old man with non-smoking history presented with L858R positive lung adenocarcinoma, given various treatments, including the first-generation kinase inhibitors, erlotinib and gefitinib, progressed 4 years after that. Biopsy revealed the classical T790M resistance mutation. Osimertinib treatment was given, resulting in a partial response with progression after 13 months. Sequencing of the progressed tumor cells showed a V600E mutation, which was not present before osimertinib resistance. So this is an evidence item describing a clinical result where all three of these variants played a role in the finding, and note how the variants are associated with different genes. So as a quick review of navigating around the new data model, I think the easiest way to get to an evidence item like this from the home screen is to go to BRAF, then load up the molecular profiles that you're shown, and click to your molecular profile of interest. On the molecular profile page, you scroll down to get to your evidence item. And of course, there can be more than one evidence item associated to a molecular profile. Now let's take a look at how we can write a few example molecular profiles. We'll start with the one we already took a look at, namely the EGFR L858R and EGFR T790M and BRAF V600E combination, which together constitute this molecular profile. The use of ANDs here is straightforward since all three of these variants are required to make the molecular profile. Next, we can look at the combination of variants underlying double-hit lymphoma, which involves a MYC rearrangement together with either a BCL2 or a BCL6 rearrangement. To write this relationship out logically, we use parentheses around the BCL2 and BCL6 variants, which are connected by OR. This allows the molecular profile to cover both MYC rearrangements with BCL2 rearrangement and MYC rearrangements with BCL6 rearrangement. And next we can look at a molecular profile that demonstrates usage of the term NOT. This is a molecular profile you could see in lung cancer patients who you might expect to be able to respond to immunotherapy treatments. These patients have PD-L1 expression for targeting with an immunotherapy treatment, but don't have these strong drivers in lung cancer like the EGFR mutation or ALK fusion. We use ANDs throughout this molecular profile since we need all three of these elements of the molecular profile to be true. NOT is placed right before the variant if you want to make sure that this variant is not part of the biology in your molecular profile. Earlier, when touring through the new interface, we took a look at this evidence item, which is associated to the molecular profile EGFR L858R and EGFR T790M and BRAF V600E. Molecular profiles are abbreviated MP for short. Recall this was a case study of a lung cancer patient, and presence of this molecular profile was associated with resistance to osimertinib. Note here how CIViC displays the molecular profile in alphabetical order. Let's take a look now and see how this evidence item was entered into CIViC using the updated curation interface that allows for the creation of molecular profiles and the entering of evidence items associated to those molecular profiles. Okay, so let's see how we enter a molecular profile-based evidence item into CIViC. We're going to go to the Add tab. We'll click that. We're going to add an evidence item. So the first thing we're going to do is start to add our variants in the form of a molecular profile. Remember, we have two types of molecular profile. We have a simple molecular profile and a complex molecular profile. The simple molecular profile consists of one variant, and this is the type of evidence that we've been entering into CIViC now since the beginning. So to proceed down this path, you would click here, specify a gene, and then after that specify a variant associated to that gene. But today we're going to be entering an evidence item which will be associated to a complex molecular profile. So we're going to click that and take a look at what we have here. This is the field where the molecular profile is going to be created, together with the ANDs and the ORs, the NOTs, and the parentheses that we can use to combine variants in the molecular profile. Let's go ahead and add the first variant to this molecular profile. The variant is going to be L858R from the gene EGFR. We click here, and let's specify the gene, which will be EGFR. After we click that, we're going to specify a variant associated to EGFR. And note that VID33 appeared here, and EGFR L858R appeared up top. We can even hover over this and see some basic EGFR L858R details. Now this VID33 is not a mistake. VID stands for variant ID, and EGFR L858R has the variant ID number 33. That indicates it's actually the 33rd variant ever created in CIViC, so it's an early variant. As you see, this was generated automatically, and you don't need to know the variant ID before going ahead and adding your variant to the molecular profile. Just as a quick note, the variant ID can be found up at the top of this in the URL for your variant of interest. Again, as you saw, the molecular profile construction interface generates these variant IDs for you automatically, so you really don't need to know this. But as a shortcut, you could go ahead and use this trick of finding the variant ID. If you are going to do it this way, make sure you're looking at the variant page and not the molecular profile page. Down here, again, this is the molecular profile page for the simple molecular profile, EGFR L858R. And if we click that, note, it actually has a different ID. Now let's return to entering our evidence item. Now what we're going to do next is connect this variant to the next one. For that, we're going to use the word AND. At this point, let's find our next variant. The next variant is going to be EGFR T790M. Again, we'll click here, find the gene, and then move to the variant. So we see now we have EGFR L858R and EGFR T790M. We will add one more AND because this is a 3-variant molecular profile. The next variant is going to be BRAF V600E. Now we have all 3 of our variants appearing. Note that they've been placed in alphabetical order. We have everything we want here, so let's go ahead and create the molecular profile. We see the molecular profile is now created, and it now exists in the CIViC database. To continue creating this evidence item, we'll move according to a procedure that should be familiar with CIViC curators who have experience making evidence items in the former interface. Really not much else has changed here, besides the fact that you now have the option to make a complex molecular profile. And recall that this simple molecular profile is a single variant associated to a single gene, so this is the type of variants that we've been creating throughout the past of CIViC and will be familiar to CIViC curators. Okay, so the next step will be to specify the source. The source is a PubMed ID, and I have the publication I'm working with right here. This is a case study describing a patient with a molecular profile consisting of those 3 mutations who has osimertinib resistance. The source has been added to the evidence item. Now I've prepared an evidence statement previously describing this case study. The variant origin is somatic. This evidence type is predictive. And the significance is going to be resistance, because we're describing a patient with osimertinib resistance. Therefore, we're choosing supports, since the evidence is going to support a role for this molecular profile in resistance. The patient was a lung cancer patient. We choose the evidence level of C because it's a case study, and the case study observed resistance to the drug osimertinib. We have no phenotypes to add. The case study seemed well described, so I will give a rating of 3. We will go ahead and submit this evidence item, and we can now view it. And here's the evidence item with the molecular profile And if we click here on any one of these gene pages, and we open up the molecular profiles, we see the molecular profile appear here. We can click it, scroll down, and find the evidence item that we just created. Lastly, let's take a quick look at some of the error messages that can appear while you're creating molecular profiles and can give you feedback on what's going on. So right here, you notice how one variant has been added, the variant number 1 actually, and behind it, the word and, which is a Boolean operator, and nothing else. So it's giving you a message that there's a trailing Boolean operator and that you need to add something else. So what happens if something nonsensical is added to the molecular profile field, like here the word fake? It gives you this feedback that what it thinks is a variant, does not match the expected format. Basically, it's telling you the only thing it would expect to see in front of a VID would be the optional word not that we've looked at before. Here you see improper syntax, where you have 3 VIDs connected by an and, then an or, but there's no proper use of parentheses. It's telling you you cannot mix and match and or like this, and that you're going to need to use parentheses properly, because a statement like this is ambiguous. For instance, it's not clear if you mean the first 2 elements or the third element, or if you mean to say the first element and the second or third elements. So in conclusion, we're excited to have this new and very wide-ranging feature added to CIViC and to its data model of how we can curate and represent the increasingly complex space of clinically significant variants in cancer. Thank you for taking the time to learn about this new feature.